So you don't give a talk. I'm going to give a talk that is sort of an overview of the HIV vaccine field and an overview of what the HIV Vaccine Trials Network does. And the HVTN is located in Seattle. It's located at the Hutch. It is really, I guess I'd have to say it's probably the NIH's largest grant. It's really a large organization, over 4 continents, having maybe 40 clinical trial sites. It does probably 80% of the clinical trials in the world in HIV. So there's an enormous number of people who I work with. Julie McElrath in Seattle, who runs our lab. Georgia Tamaros at Duke, who runs the Duke lab that does the binding antibodies. And we thank our funders, both the NIH and the Gates Foundation. So we work with a lot of companies, and so I want to acknowledge a lot of people who are participated in this. So the structure of most talks is to sort of tell you why you should listen, the first part. And the first part is, HIV is still a global problem. In fact, we have sort of HIV fatigue. When I look at all the stuff that happens over Ebola that has 3,000 deaths a year, and we still have 2 million HIV deaths a year, and 2 million acquisitions a year. So it's still a problem in the United States. These data here are, if you just look at the axis and you add these up, there are about 50,000 cases, new cases of HIV infections per year in the United States. And it's been steady despite us having antiretroviral therapy for a long period of time. And the reason for that is that one of the themes is that you can't control a sexually transmitted disease that is acquired asymptomatically, that is transmitted asymptomatically, that is essentially asymptomatic in its transmission without a vaccine. We will never do that without a vaccine. And the data here are that. If you actually look at what is called the continuum of care, about 85% we think of the population that have HIV is diagnosed, and the whole emphasis of doing HIV testing routinely in hospitals and medical care facilities to to essentially test and treat. But even if there's 85 to 90% that are known, only 40% at the moment are treated or actually in medical care. Only, let's say most in medical care are getting treated, but only about 75% of them, or over 30% of the population, are essentially suppressed to levels that would decrease the transmission. So there's the rub as it relates to the issue of test and treat. We still have microepidemics. There is an epidemic of amphetamine abuse and at times heroin abuse that continues in our country as we don't deal with the medicalization of IV drug abuse. And even in rural Indiana, in Scott County, there was an outbreak that led to essentially 155 cases of HIV, all derived from one person and sharing needles. And if one saw the recent HBO documentary on HIV, you can sort of see that average needle sharing per episode in rural Indiana was, shooting up with 8 people at a time, and sharing the needles with 8 people at a time, and leading to this essentially very homogeneous outbreak. HIV is not a trivial disease, and if you actually now, I started my career in antiretrovirals, and I ran the AIDS clinical trials group when we first showed AZT, then a DDI, and then we put all 3 drugs together, and we changed the nature of this from bringing a person to teach the medical school class and then dying every year. And when Bob, who was my fellow then, started the clinic here, when we actually now look at the fact that the average duration of someone on triple therapy is about 40 or 45 years. But there's still an immune activation syndrome, even with suppression, and there still is an increased death rate, and the death rate is for cancer, what is called non-AIDS cancers, but in my opinion, it's AIDS because it has aspects of inflammatory reactivity that is associated with increased risk in cancer. So cancer, heart disease, drug overdose, suicide, liver disease. These are sort of the major causes of death in the United States. I think the issue with cancer and heart disease really portend amazingly for the global health issues. When you look globally, in the world now, there are almost 40 million people living with AIDS. So again, these are numbing statistics. When you look at new HIV infections, there's 2 million new HIV infections per year globally. If you look at deaths, yes, we are starting to treat and change the mortality. And in some countries like South Africa, that had decreased its mean lifespan from the upper 60s to almost to the mid 50s, where 30% of the deaths are HIV, and another 15% of the deaths are TB, and 80% of those deaths are HIV associated. We're now sort of seeing reversions of that. So we are seeing AIDS-related deaths fall. But it's still a numbing statistic here. It's 1.3 million deaths from HIV per year. So this is numbing. Now people want to talk about an AIDS-free generation. That's sort of the goal here at the moment. And what does it really mean? When you look at prevention opportunities, there are a number of sort of tools. Obviously, condoms, abstinence, but there are a bunch of antiretrovirals that have been out there. Pre-exposure prophylaxis and post-exposure prophylaxis are the ones that have been used, and of course, circumcision. And frankly, prevention trials have actually shown that they can work. But the reality is that they work markedly associated with their adherence, and their adherence decreases over time. We haven't been able to do in infectious disease what the cardiologists have done. They make high cholesterol a disease. We haven't actually been able to say that getting HIV is a disease, and compliance associated with that is really lousy. And on a population basis, there is no evidence that PrEP can decrease on a population basis HIV acquisition. It is a great case management tool. Maybe test and treat on a population basis could actually do that if you really get everybody suppressed. But on a population basis, we have no tool to decrease that. And to me, an AIDS-free generation means you grow up. I want my granddaughters. I got 5 granddaughters and a grandson, so I'll say granddaughters and grandson. No inequality when it comes to sexuality and responsibility. That they are able to grow up like I grew up, without a fear of AIDS. Now, I grew up because before HIV was recognized as a disease or an infection, existed, but obviously in Africa, not recognized. So to me, an AIDS-free generation means you get the cases like we have polio or measles or mumps. We would go from 50,000 to less than 1,000. I think test and treat and PrEP would probably get us from 50,000 to 20,000, and that's an incredibly useful thing. 30,000 lives prevented per year. That's not trivial. But really, to actually make this an AIDS-free generation, you really need to make an HIV vaccine. And I think from a global health point of view and an economic point of view, we cannot test and treat ourselves out of this epidemic. The only way we're really going to solve it is to develop a vaccine. And I would say I'm not sure that there's a problem on Earth in healthcare that is more important globally than to essentially come up with an HIV vaccine. Now, it has been a lot more difficult than I imagined. And I can look back and say, okay, I've spent 15 years in the HIV vaccine field, and I'm very grateful that I started my career in therapy because success has been, a little bit hard to take. So the outline of the talk, I'm going to be a short overview history, the current state of HIV vaccine development, which I sort of say we're at a Yogi Berra moment, which is when you come to a fork in the road, take it. We're learning about the fact that antibodies are probably what actually controls HIV. Even as a T-cell guy, I went down that rabbit hole, and I'll explain that. It was a bad rabbit hole. Not a bad rabbit hole, but didn't lead to the promised land. And the recognition that non-neutralizing antibodies may play a bigger role than we've given credit to. talking about the ability to look at neutralizing antibodies. And then dear to my heart, because this is what I work in the moment, and mainly in genital herpes, is to use tissue-resident memory T cells and their role in clearing infection. So the major questions facing the HIV vaccine field are, can we induce vaccine regimens that give non-neutralizing antibodies to an effective enough titer to actually be effective? Almost all vaccines that we have, I would say essentially all of them, that we think work by an antibody-mediated mechanism make neutralizing antibodies. And that has been the holy grail in HIV. Now the problem is that, as I'll show to you, is that essentially HIV is the HIV envelope is a uniquely poor immunogen. And it took us about a few years to recognize it, but essentially when you immunize with any sort of envelope antigen that is made by recombinant technology at this point in time, you make great neutralizing antibodies to the antigen itself. You hardly make any neutralizing antibodies to the brothers and sisters. You certainly don't to the first cousins, and you never actually do to the neighborhood. So it's uniquely narrow and actually problematic from that. And the second issue is even if we can make neutralizable antibodies and an immunogen, is it associated with vaccine protection and will be of significant magnitude to overshadow other design approaches? What's the role that CD4 T cells play in their interaction with B cells in making antibodies? And if we concentrate on the CD4 T cell response and increase the T follicular responses that are the T helper cells that help the B cell, can we sort of alter that machination and interaction between those two cells to improve the antibody responses? Or are we just going to increase the activation? We actually have data in which the stronger vectors have done worse with respect to protection, because we actually may deposit more CD4 cells that are activated into the mucosa and essentially carry in more activation into the genital tract and actually, increase the likelihood of acquisition. And we have this yin and yang of how do we make more antibodies with not more activation, and maybe a quiet vector, and that's the reason why this canarypox vector program is looking good. We thought it was the most unimmunogenic and sort of uninteresting vector, and it seems to be the most successful vector. So it's teaching us that there's a yin and yang, especially with this virus, as it relates to the role of activation, especially CD4 activation, which of course is the target cell of HIV. So the stages of HIV vaccine development started right after the virus was isolated. We had Secretary Heckler said, hey, we'll have a vaccine in a year. I got our first grant here in HIV vaccines in about 1987, 88, 86, when they started putting money into HIV vaccine development. And the first generation approach was, just follow hepatitis B, make a recombinant glycoprotein that was on the surface protein. And we, lots of companies were started. Wall Street poured in money. We made GP120s, GP140s, 145s, 160 is the whole thing. They were all immunogenic. They all gave strain-specific neutralization. They gave you what we call binding antibodies, which at that time we thought was really schlock. They gave us ADCC antibodies. I've been working on ADC and herpes. Now I'm actually reinventing ADCC. I'm going back and redoing the assays that I did 25 years ago. We actually didn't think much of it at that point in time. There were 2 large efficacy trials of GP120, and they were duds. They did absolutely no activity. So there was a frustration that a recombinant protein could not be put into a structure that induces broad neutralization, and teams involved in this work essentially were dismantled. There was at least 8 companies that went belly up. There was a large-scale dropout of the pharmaceutical and biotechnology companies from HIV vaccine field, and there's essentially no company now that uses its own money to make an HIV vaccine. Any money or floor space that a company uses comes from public-private partnerships in which they're getting money from either the Gates Foundation or the NIH to use their personnel to do that, and that's a good thing. I'll come back to that. Wall Street became very wary of financing HIV vaccines, in fact, like any vaccine. And there's a recognition by the government that philanthropy, that funding for vaccines must come from them, and that we actually needed to develop the structures in academia to develop products. And that's sort of when we came out and reorganized this thing and called the Global Vaccine Efficacy, and we started putting together these product development companies within academia, and that's been pretty successful. Now, my comment on this is that inducing envelope antibodies is not necessarily a bad idea. It's just that making monomeric GP120 is not the right execution of that. And that HIV-1 envelope is a uniquely difficult immunogen. It induces poor durability. It has poor induction of conformational neutralizing antibodies. We've been unable to actually make the trimer, which is the active form, and the virus itself has less trimers on it than any other virus. So the whole issue of actually presenting a particle that actually has like 120 things on it, has never, the virus itself, one of its immune evasion strategies is to put less trimers on it than actually any other virus that we're aware of. Now, the second generation vaccines became the T-cell based vaccines, and that sort of came from two rationales. Number one is that the CD8 toxic T-cells were induced early after infection. B, long-term non-progressors seem to control it through their T-cell response. And three, the recognition that the T-cell responses were to essentially more conserved regions of the HIV genome than to the envelope, and that the envelope protein was much more diverse in its conformational epitopes and its glycosylation patterns, and was very plastic as it relates to the glycosylation patterns, but the conserved regions were there in which were to T-cell epitopes. And so the field essentially shifted totally to vaccines that induced CD8 T-cell responses. And we did a test of concept trial in which we took a very potent replication competent adenovector. It was the adenovector type 5 that had just the internal structural genes, GAG, NEF, and POL, and did a vaccine study, and which was called the STEP study. And to our dismay, and frankly, a very tough time for me personally, was we showed an increased rate of acquisition of HIV. The mechanism of still, which is unclear, we actually do think we induced more CD4 positive T cells, and it was mainly in a subset of men of uncircumcised gay men. The mechanism really has been unclear, but it certainly has been a sobering issue for the field. Shortly after that, there was what was the great surprise of a trial that was started by the military and took this vector called the ALVAC vector, a canary pox vector, and sort of my own career. If you go back, the very first studies of the canary pox vector I'm the first author on. And I had sort of the choice of the HVTN to do the efficacy trial with the adenovirus vector that was made by Merck, or the ALVAC vector that sort of went to the military and the ties. And I picked the adenoviral vector, not the tie vector. The tie vector was being put with GP120, and I looked at the tie vector, and it didn't have very much potency. And it was also put with a GP120 that already had shown not to work. And I sort of looked at that and say, well, how could this weak vector with this protein that already didn't work? Why would I want to do that? And so, but we wanted to make sure it got tested, and it went to Thailand, and they did a wonderful study. And the reality is it did work. The curve separated really quite early. It had about 60% efficacy in the first year, and then it changed and decreased over time to overall about 31% efficacy. And there was then this sort of massive effort to sort of say, well, wait a minute, how did this work? How could you take something that was sort of half of it already been shown not to work, add just this sort of vector and figure out what went on here? Here is where an international group of scientists came together and provided some really very critical insights. First of all, it was clear it was not neutralizing antibodies. When they looked at the isolates and looked at neutralization, the vaccine did not induce neutralizing antibodies to the circulating isolates. And the tie epidemic is really actually was a good place to do it because it had a less diverse genetic aspect of that epidemic, was really pretty concentrated in the strains that were present. when you look at the immune response, it induced very good immune responses, peak immune responses. The regimen was 0, 1, 3, and 6. And up here, there were a lot of binding antibody responses. We'll come back to this. These are all non-neutralizing antibody responses. They had a lot of what we call antibody cell-mediated cytotoxic responses. So they're functional antibodies, binding antibodies. They waned over time, and this waning over time is associated with decreased efficacy. And there was then a large sort of biostatistical and laboratory program to sort of look at, are there antibodies and or T-cell functions that we could define that separated people who actually were protected versus non-protected? Are there antibodies that are correlated with that? And our surprise of that is that, yes, there were. And some of these related to a region of the virus that, frankly, we thought so little about and actually had largely been omitted and cut out from almost half of the GP120s and GP140s that were made earlier on. I mean, what we called the V1, V2 loop. You know, these were sort of felt to mask the V3 region, which was felt to be the immunodominant region. A lot of the things were clipped out of there. And it turns out that this vaccine induced a lot of V1, V2 antibodies, both picked up by linear epitopes and then by conformational epitopes. And then I'm going to show you the story that people who had the highest titers of antibody to those V1, V2 loops had as much as 70 or 80% protection. So the reality is that a lot of the primate models, the non-human primate models, the rabbit and other kinds of models, you know, were not predictive of this, because those V1, V2 deletions actually gave you higher neutralizing antibody titers, higher binding antibody titers, but in the immune response, that's not occurred. So antibodies to the conserved region of V2, previously almost completely ignored, were highly correlated with efficacy. Also, the CD4 T cell responses, especially the CD4 T cells that functionally made cytokines that actually affected B cell development. I'll show you that the polyfunctionality was really as related to IL-10, IL-13, CD40 ligand, that those polyfunctional CD4 T cells were important in protection. There was no CD8 response to this vaccine, and there was no significant neutralizing antibodies. So associated with vaccine efficacy was high binding antibodies, the avidity of the antibody, the V1, V2 antibody, the ADCC activity, and the magnitude of the CD4 T cell response. Just to give you a flavor of this, these are the V1, V2 antibodies, and this is the people who had baby bear, mama bear, and papa bear kind of responses. This is their tertiles, and these are the Kaplan-Meier curves to show that those who had the highest and medium had significant reductions in the probabilities, and that's about a 0.68 probability. And actually, if you look at this, which is the estimated vaccine efficacy, looking at the immune response to V1, V2, and this is a histogram of where the tertiles are, that this is a pretty linear relationship. Now, this is only one study, but my dream is that if you could push this entire curve over to the right and make a regimen that doubled the V1, V2 antibodies, you would continue to move this up into the 70 or 80% efficacy. Now, these V1, V2 antibodies were functional. So they weren't just binding antibodies, but they mediated ADCC activity. When you isolated monoclonal antibodies from the people who got vaccinated and you looked at those monoclonal antibodies that attached to V1, V2, they actually mediated ADCC activity at a couple points in time, at a couple places. They also captured virions, and they actually synergized with a bunch of other monoclonal antibodies. Now, the other data that made us a believer of this was that one did a genetic analysis of the envelope sequences of those who got vaccinated and were protected versus those who were vaccinated and placebo recipients who acquired infection, and looked at the genetic makeup of the envelope, and especially around the V1, V2 region. And one found that two sites, one at site 169, one at 81, in which there was a mismatch between the placebo patients and those who had vaccine. And that's shown in this graph when you had a K at number 169, at lesion to lysine at 169, and you look at the, the incidence of those who had placebo versus those who got vaccine. So the people who got vaccine actually showed pressure on the viruses that had this K at 169, which is at the peak of where the binding site is of the V1, V2 loop. So when the genetic sequencing data from the virus, which is sort of unbiased, begins to match the immune profiling from the vaccination, which you could say is potentially biased because it's a case control study, one starts to become cautiously optimistic there's a modicum of truth there. And that's sort of the breakthrough feeling about non-neutralizing antibodies. Second question that had to be asking was, what did the vector do? We already gave GP120 and showed it didn't work. So what was the different immune response that ALVAC, this canary pox, did that was different? And there the data have been sort of softer. Did it enhance innate immune responses? The curves separated right away. Were there differences in antibody responses, and were there differences in adaptive T-cell responses? Of course, there were for sure in the last one. Now, ALVAC priming shifted the immunotype or the antibody switching of the antibody response to the GP120. And there were much more high IgG3s than IgG2 and 4. There is some literature that IgG3 in humans binds to and is more functionally active in binding and in even neutralizing viruses, so there's precedent for this. And persons with high IgG3 responses had higher ADCC activity. The problem is, and also the fact that it induced these IgG3 responses only transiently. So it was 80% at the peak response, and it markedly went away over time, which goes away with the, which correlates with the waning immunity. So when you look at the binding antibody responses in those who got ALVAC vector versus the old trial in which you got GP120, you see that there's big differences in the V3 responses. Those who get the antibody alone had very high levels of V2 and V4 as compared to the ALVAC prime. There was another mechanism that HIV uses that we're beginning to sort of understand in the vaccine field and understand actually in the virus's pathogenesis, and that is in blocking or distractor antibodies. And we've identified sort of two functional distractor antibodies. One of them is GP41, or part of GP41, which is essentially a cross-reacting antibody between the virus and E. coli ribonucleotide reductase, which we all have in our gut. And we all have B-cell priming too. And one of the things that we've recognized when you actually get natural HIV infection, GP41 is the first antibody made. And when we vaccinate people, GP41 was the first antibody made. But that antibody actually blocks. That antibody actually makes a B-cell repertoire that essentially is directed at the envelope protein, but essentially prevents a very large repertoire of GP120 to occur. And we've given some vaccines that actually do that. And the other aspect of that is that there are serum IgA antibodies which actually induce immune responses that actually, by their FC receptor, bind to the C1 region of GP120 and block binding and effector functions of IgG, including ADCC activity, and essentially block ADCC activity. And we actually showed that the people with the highest titers of IgA actually did worse than those who had low titers of IgA. And the people with high titers of IgA had less ADCC activity. So how do these strategies teach us? They teach us a couple things. Not every vaccine does this. There are some vaccines in which we make a lot of V1 and V2. There are some vaccines in which we make a lot of blocking antibody. And there's some vaccines that we can make antibody to GP41 that has an immunodominant region that may be functionally important. So we learn in science that the quality and the epitope-specific responses. And so now we actually have a program in which you're sort of trying to take away and create an immunogen that doesn't make the blocking antibodies and get us closer. The last thing we learned from the trial is that as I talked to before, that CD4 T cells that made CD40 ligand, IL-2 and IL-4, if you did that, your relative risk was a 62% efficacy, and that was associated with greater protection. So how do these data lead to better next steps for HIV vaccines? That's really the issue here. How do we take non-neutralizing antibody functions and lead to a better vaccine efficacy by giving better binding antibodies, by giving higher binding antibodies to HIV envelope, to V1, V2, to better CD4 responses? And we now have sort of 2 clinical trials that are starting, that are testing these hypotheses. One of them comes from the consortium of people that looked at the RV144 trial and said, what do we do next? And basically what we decided to do was to reconstruct the ALVAC vector, take the GP120, direct it towards where the epidemic was needed the most in sub-Saharan Africa, and construct clade C sort of counterparts of this. And take a look at the issue of increasing the immunogenicity by making the vector a little bit more sort of potent, as well as by adding a more potent adjuvant and a better boost. And that is a trial that actually, it took 6 years to make these products. I'm not proud of that, but it's not my fault. Companies take a while to come to consensus to pick the strains and get them to be manufactured. We've just about to finish the qualifying trial, and we think we will start the trial to sort of see if this approach will work in the fall of this year. This is the observed immune response that we saw in RV144. It went up and went down. We are expecting that what we will do is achieve this response with the clade C by boosting it and using a better adjuvant. We will actually continue this, and we will hope that the efficacy remains in the 60 or 70% regimen during that period of time. And that is happening. The next approach has been one that actually takes a look at improving the T cell responses, the CD4 T cell responses. And that is an approach that was initially started at Harvard and picked up by Janssen Pharmaceuticals, and they have been a wonderful partner. And they are using ad vectors. They are using what we call a mosaic antigen, which is essentially a synthetic envelope that has sort of is based upon to try and cover in one vaccine all the clades. They have made a trimer, a GP140 trimer. And in non-human primates, this vaccine has actually given better protection than any other vaccine that we've seen. This is just a non-human primate study that actually shows that if you give the vector plus the envelope alone, the vector itself gives a very good CD8 response, and the vector itself gives a reasonable CD4 response. But if you want CD4 cells and antibodies, you give the vector plus the envelope that increases the CD4 response. And if you look at the protection, if you give the envelope alone versus the ad envelope, you get increased protection by giving the envelope. You get markedly higher binding antibody titers. And what correlates with this is not neutralization, but ADCC activity. ADCP activity is essentially ADC activity using monocytes or macrophages. It's antibody-dependent cell-mediated phagocytosis, and this is a neutrophil assay. So essentially non-neutralizing effector functions are what is being thought about. So we have 2 approaches to look at non-neutralizing antibodies. Now, how about neutralizing antibodies? At the moment, we do not have a neutralizing an imaging that gives very good neutralizing antibodies. But out of the technologies of B cell, of isolating B cells and screening for broadly neutralizing antibodies of the last few years, there have been a plethora of broadly neutralizing antibodies isolated from HIV-infected persons. About 10 to 15% of HIV-infected persons have broadly neutralizing antibodies. Frankly, most of those persons have progressive disease. It's not like hepatitis B or hepatitis C, where if you develop those neutralizing antibodies, you actually clear it. These antibodies arise in mostly in people who still have progressive disease, who are seeing a lot of viral antigen. But the reality is that when you take these antibodies and you look at them in vitro or in vivo or in non-human primates, they block acquisition. And we are able to now make those antibodies in high titers, and we're starting a study in which we're actually passively infusing those antibodies to sort of see how well are neutralizing antibodies going to be effective in HIV prevention. So you can use antibodies for many things. When you look at the neutralizing antibodies to HIV, they've been directed to many sites, but the ones that are in particular that advance in the clinic are to the CD4 binding site. That is the attachment site of the virus. And if you look at those antibodies, they block. Now, a dogma that I've taught all the fellows, I believe myself, is that neutralization is the holy grail, and that everybody in the HIV vaccine field feels that we get neutralizing antibodies if they should work, if they're broadly reactive. Passive transfer studies in non-human primates and the accuracy and reproducibility of neutralizing antibody assays in world-class laboratories have been done. Now, I myself in my career have, however, worked on 2 viruses, made vaccines that induce neutralizing antibodies, and they haven't worked. And so I'm a little bit more of a skeptic from my own experience. I worked with Merck as well as in GSK in making a recombinant protein vaccine to HSV2. They made levels of neutralizing antibodies that we would kill for in the HIV field, and we've flushed out with that. There have been neutralizing antibodies to CMV, glycoprotein B, which gives neutralization, and that has not worked very well either. And there's been a recognition that actually in the CMV field that you can, a different structure, there's now a pentameric structure. The monomeric GB makes one kind of neutralizing antibody, but there's a pentameric structure on the virus that actually makes neutralization that blocks, the GB blocks the virus in epithelial cells, but the pentameric structure blocks it in myeloid cells and in neutrophils. And essentially that's needed in order to actually prevent transmission. And so you essentially need to validate your neutralizing antibody assay. And so that's what we're doing now. There's a monoclonal antibody that was isolated at the NIH, and it's really amazing that you can take a monoclonal antibody that's first isolated from a human in 2011, get it into the clinic so that you can do a phase 1 study by 2014, and we're going to start an efficacy study that is with 4,000 people giving 42,000 IV infusions to test this antibody for HIV acquisition. And that study is starting next week in New York and in next month in South Africa. And I'll explain that. And it's an amazing kind of accomplishment of the infrastructure that has been built in HIV, the infrastructure that has been built in the public sector with public funds to solve some of these things. And it's manifested itself why vaccines for Ebola and Zika and all these other kinds of things are now possible, and what can be done, and where HIV has sort of driven the alteration in thinking about the infrastructures that's required to deal with rapid public health things. And people who think about, gee, we should alter the HIV funding, you should understand as to just how unbelievably we've changed the nature of product culture in nonprofits, in the university, in this university or at the Hutch, probably more at the Hutch than here, with respect to just the culture that exists. And we'll talk about that off record. So how potent is VRC01? Number one is that it's able to in vitro neutralize almost all the clade viruses. The percent is here at less than 50 micrograms, and a very high percent at less than 1% micrograms. So it is broadly neutralizing. The mean IC80 is 1 microgram, so it clearly works at physiological attainable levels. It is actually seen in mucosa and is actually actively transported by the neonatal FcRn receptor, the neonatal Fc receptor. And if you give it to a non-human primate and biopsy it, you can actually see very high concentrations in rectal biopsies and rectal mucosa. And that's actually, this is staining for the antibody itself in rectal biopsies. Now, the mechanism of protection of monoclonals includes the FAB end, the neutralizing end, but it also has an FC end. And one of the issues that it looks out there is to sort of say is, you know, what is the concentration that's going to work? Is it going to be predicted by the neutralizing antibody assay, which is just an in vitro neutralization without cells? Or is it actually going to work better in vivo when you have cells and it has an FC part of it? Because it will have ADCC activity. It will have binding activity. It has ADCP activity. And, you know, will we be, quote, lucky and be able to show that lower concentrations are effective? Will all escape breakthroughs be neutralization resistant? Or will we actually be able to protect against some neutralization-resistant isolates or relatively neutralization isolates by giving this infusion? So A, will it work at all? How well will it work? And what effector functions are associated with that? In non-human primates, you can give the antibody and then challenge it with what is called a SHIV, which is an SIV virus that has been, you know, a chimera in which you've put an HIV envelope. Now you can, you know, any good investigator knows that you can, you know, rig your experiment to however you want it to be. So you can do different kinds of SHIVs. So there are SHIVs that are resistant, and there are SHIVs that are very sensitive. So you can put an envelope out there that is relatively resistant and shove it into the macaque, and you can put a sensitive one, and you got to understand the literature when you look at different things. So insensitive ones down here, the SHIV-BAL4, you know, very low concentrations of antibody, you know, work. You want to give it higher, you're going to get less efficacy. So this one is actually in a relatively higher one in a rectal challenge. If you give 20 milligrams per kilogram, you're totally protected by rectal challenge, and you're partially protected by vaginal challenge. Here's a comparison between VRC01. This is a monoclonal antibody to the V1, V2 loop. This is another, this is the, to the membrane proximal region, and this is a control. And, you know, you can see the VRC01 does pretty well until you get to pretty low levels. It looks better than PG9, but I could put in something that makes that PG9 was, you know, more potent too, and I could shift what these graphs look like. So the reality is that, you know, monoclonals look good, but you don't know what they are until you put it in humans. So we are starting what we call antibody-mediated prevention. And we like these mnemonics, AMP. So we're amped up for the study. So it's really, can a passively infused monoclonal antibody prevent HIV acquisition in high-risk adults? We've done a phase 1 study in which we've given 40 milligrams per kilogram, 10 milligrams per kilogram, and 30 milligrams per kilogram. Let's say the ones that we're interested in, which are the 30 milligrams and the 10 milligrams, you get levels up to 300 micrograms per ml after infusion, and the trough level is at 30 micrograms. And I'll tell you the average IC80 is 1 microgram. So we're above lots of antibody for all 8 weeks that we're going to do this. At lower levels, which is 10 milligrams per kilogram, the peak levels are at 100, and our trough levels are around 8 or 10. I will say if that we get protection here, there's a chance that we could make an immunogen that does it at this level. If we only get protection at 30 milligrams per kilogram, I'm not sure there's a protein immunogen that we could conceivably think of making at this point in time that would induce this level of neutralization. So this is a study that we're looking at. Not only could monoclonal antibodies be used, you know, chronically over time, but it will set the stage as to what is possible from a, you know, a vaccine development point of view for the next decade. And that's the reason to do this kind of study. So it's a placebo-controlled trial. We're taking 2,400 MSMs or transgender women in North and South America and randomizing them into the 2 doses versus placebo. They get infusions every 2 months. So it's at every 8 weeks, and we do that for 20 months. We're taking 1,500 women in sub-Saharan Africa, and we are in South Africa, Malawi, Mozambique, Tanzania, Kenya, Zimbabwe, and Zaire. So we feel very comfortable in our sites that we can do these IV infusions at the site. The study's powered to detect 60% efficacy. We've had very good safety in our phase 1 study, and that's opening next month. So the scheme of the trial is, this every 8 weeks times 10 doses, every 8 weeks times 10 doses. I don't think a study of this magnitude has ever been done of an IV infusion over that period of time. We've spent a lot of time with the Japanese lean thinking. Paul Strangert would be really happy to know that I was a great student of his lectures. We hired consultants, we've done things. We've gone through our clinic. When we first started the phase 1 studies, even in the United States, it was taking 4 hours per clinic visit. We came in and did the rapid processing development, and that visit now is a streamlined 75 minutes. From the time they hit, we have a video. This is how you do it. The infusion is a half hour of that 75 minutes, and they're in and out. And this goes to show you what you can do if you put your mind to it. So it is to inform future HIV vaccine design because we are now starting to be able to make envelope proteins that look like native-like envelope trimers. We're starting to put those in humans. Even in non-human primates, we see levels that are sort of comparable to the kind of 1, 2, 3, 4 microgram levels that we see of the monoclonal antibody. If it requires high levels, then I would have to say that if monoclonal antibodies have really, are a huge business, both for rheumatologic disease and cancer, and the ability to manufacture those, increase their half-life, develop strategies to deliver it in a longer way. Vectors, whether it could be an AAV vector that could be given once and be there for 8 or 10 years, or put it in a plasmid that are now plasmids or B cells, autologous B cells that could go in to give you long-lasting antibodies. So conceptually, the delivery systems are being developed that if we show that it requires high levels or persistent levels, I think we could actually make this into a useful product. It also has a potential role in interruption of maternal to child transmission, still a problem during breastfeeding. And it's been shown to work in neonatal macaques. So we need a proof of concept in humans and that we can essentially re-engineer both the delivery system and the antibodies per se. I'll end to say the last concept is that we are seeing T cells revisited. There is a CMV vector that is a unique vector that has a deletion of those, essentially of the pentameric neutralizing antibody structure that is capable in non-human primates to actually essentially clear HIV infection, or it's been an SIV infection in about 50% of animals. And this vector itself gives a unique T cell breadth. It gives a unique profile of T cells. We get class 1 restricted CD4 T cells and class 2 restricted CD8 T cells, and that much of the response is to HLA-E. Now, all of us who have been looked at this sort of look at that and squint and say, what the hell is that? Well, HLA-E is probably our primordial HLA type. It's a very promiscuous HLA type that probably was one of the first HLAs that we first had, and we evolved maybe, very good on a population basis, but maybe not less good on a person basis. But if you look at the data here, which is in an extreme SIV challenge that has never been cured before, about half the animals who receive vaccine essentially are down here being cured. And actually, when you take the blood from these animals and transmit it to other macaques, they look like they actually been sterilized. If you look at the immune response, and this is the immune response to GAG, in a human immunized with almost any other vector, you will get 1 or 2 or 3 epitopes. In a macaque, which has many more MHC types, you will get 5 or 6. And here they're getting an epitope essentially every 7 or 8 amino acids. They're getting like 44 epitopes. And most of these are HLA-E restricted. And it is a very unusual and interesting and response that is asking us to sort of rethink some of the basic immunology that we've learned over time. But it's a pretty exciting concept. The issue is, can it be duplicated in humans? If you make the same deletions of human CMV, will you actually sort of see this alteration? We think this alteration is due to antigen processing, that there's something going on with respect to antigen processing that is herefore not be recognized. And we'll be able to use this. This is a vector that doesn't make antibodies. It just makes T cells, but it makes T cells that seem to be effector T cells that persist in tissue for a long period of time. It's an interesting story that I'm just sort of giving you a smattering to sort of highlight and think about, and sort of, I would say from a biology point of view, those who are interested in immunology, to sort of look at and watch and see how it develops. So there's considerable energy, I would say, in the HIV vaccine field. We're initiating some test of concept studies of both neutralizing and non-neutralizing antibody approaches. I think that will set the stage for the entire design and development field for the next decade. I think for the first time, we're able to do things in humans that will, I think, drive the basic science agenda, because frankly, the small animal models and the non-human primate models have not been predictive, and we only learn by going into humans. And I have some cautious optimism that our goal of achieving an HIV vaccine that can significantly reduce HIV acquisition and start the reality of achieving an AIDS-free generation is possible, and maybe I'll get to see it before I retire. Thanks. And again, the huge number of people that generated the data that come in these slides.
Thank you for a beautiful talk. I think there might very well be some questions. And let me start out with one. Since antibodies are back in, at least to some extent, and IgG3 in particular, one of the features about IgG3 is it tends to be better in activating complement than the other IgG subclasses. Is there any role for complement activation in protection from HIV?
Well, I think there's been precious little look at it. I mean, all the, essentially all the neutralizing antibody assays are done without complement. So whether complement-dependent neutralization is there, buried into this, there's an investigator by the name of Khalid Alter at Harvard who has developed a series of binding assays that look at with and without complement and are sort of in their systems vaccinology. And one of the ADCD assay is a complement-dependent assay, which correlated actually in the non-human primate with non-neutralizing antibody efficacy. So I think your comment is spot on, and we need to actually do a better job of looking at that. Yeah.
What has modeling told us about the minimum efficacy that would make a dent in high prevalence areas, and what's the political reality of actually using a vaccine that didn't have the objective of providing complete protection?
Okay, so the question is, what's the reality of a licensable vaccine efficacy? What's a licensable vaccine efficacy that modeling studies have done? So this has been done and discussed with what is called the Medicine Control Council in South Africa, which is the country that has the most HIV-infected persons, and where we're doing this study. And the modeling suggested that we would save an enormous amount of lives at a 50% efficacy. And so that is the goal that we've set both at the trial to have a 50% efficacy over a 2-year period of time. We would hope that we would get increased durability. As you would know, when you actually just intuitively think about effect of vaccines, that durability is probably the single greatest. You need to have efficacy, but if that efficacy has to be with durability. So, we're looking at 2 years as the minimum durability, because that actually was what the original model is, and there's the hope that with boosting, you could increase that durability over time. So we'll see where that takes us, but essentially 50% is sort of where we're at. Now, whether the US would accept a 50% efficacy in modeling and our epidemic would be 50%, we'd have a lot of controversy over a 50% effective vaccine in our country. And we haven't touched the FDA yet with respect to what that would show or how we would feel and where that would fit in the mix of tools and where the economic analysis. Big difference in economic analysis between where the general population is 7 or 8%, and acquisition rates are 20% in teenage girls versus what we see in the United States. Yeah. Bob.
Larry, does your modeling include the use of PrEP? Because if you combine that with even a 50% vaccine efficacy, you could be looking at something in excess of 90%.
Right.
Even with less than optimal compliance with PrEP.
Yeah, so Bob raises, so the point Bob makes for the TV audience is the issue that a partially effective vaccine with a partially effective antiretroviral approach, whether that's daily oral PrEP or what is now happening with injectable PrEP, that we could, if you put those two together, 50 plus 50 is close to 100. There's no reason to believe that it wouldn't be additive. And I think that's where the field is that if we get at 50% or 60%, you'd look at it with respect to enhancing the current prevention package and get us to really where we would be talking about an AIDS-free generation with a combination toolkit. And I think the point is a beautiful one and exactly spot on.
But it's also going to make the issue of testing your vaccine difficult because the use of PrEP is going to, I don't want to say contaminate, but it's going to be a reality. And does that mean larger studies than
Okay, so Bob again astutely asked the question of, how does the use of PrEP affect our vaccine studies? Well, in the United States already with the trials that are ongoing, we are offering PrEP. And anybody who wants to be PrEP, we have a program in which there's Gilead, the maker of Truvada, tenofovir FTC, that is licensed for pre-exposure prophylaxis, what we call PrEP, is donating the drug to, we have a mail order pharmacy. And so anybody who has access, we provide them referral through their physician to get this program. I mean, the reality is that unfortunately, unfortunately, that there is a lot of resistance to taking PrEP. There are a lot of people who, even when counseled to do so, don't take PrEP. And if they take it, they don't take it reliably. So we inform people about PrEP. We make access to PrEP available in the trials. As I said, I came from the antiretroviral field, and I believe in antiretrovirals and antivirals per se. If it does decrease the acquisition rates, and we have to make the studies larger and they end up being more expensive, well, that's what clinical research is all about. So we do monitor the use of PrEP during the study. We do that both objectively as well as by questionnaire, and the DSMB that controls the study will be monitoring that. And we have to add people or alter the study design in some way, we will.
Well, there may be additional questions, but I think in the interest of time, we should end now. But again, thank you very much for allowing us to also be optimistic about studying this epidemic and maybe getting it under control.
Well, my pleasure to be home, and thank you for all.
Thank you.